Good afternoon and welcome to the annual Founders Day Faculty Science Lecture. I would like to acknowledge all our founding faculty who are joining us today, albeit virtually. Each November, we gather to remember the founding of the College of Medicine Tucson. The dedication ceremony for the College of Medicine took place more than 50 years ago on November 17, 1967. The focal point of the event was a ribbon cutting by then University of Arizona President Richard Harville and Arizona Board of Regents President George Chambers. What started as a single basic science building, 32 medical students, a handful of faculty, and a teaching hospital that opened its doors in September 1971 has since grown to become a significant source of pride and hope for the people of Arizona. Today, the College of Medicine Tucson provides a wide variety of health-related academic and clinical programs locally, regionally, and nationally. Since our establishment, hundreds of College of Medicine Tucson faculty have contributed to the education of more than 4,000 physician alumni. This includes several of the college's founding faculty. This annual celebration and the Founders Day Lecture provide the opportunity for us to reflect upon our history, our many accomplishments, as well as providing us the opportunity to recognize those individuals who have contributed so impactfully to the college's growth over the years. The Founders Day Lectureship was established in 1979 to recognize college faculty for their scientific contributions. Nominations for the Founders Day Lecturer are solicited annually. Faculty and staff are invited to nominate peers, but not only for their contributions, but also for their ability to inspire others. Thus, the Founders Day speaker represents the college's recognition and acknowledgement of individuals held in the highest esteem by their peers for their scientific and academic achievements. Today, in continuation of this tradition, I am honored to introduce our Founders Day speaker, Dr. Marvin Slepian. Dr. Slepian obtained a bachelor's degree in biochemical sciences from Princeton University, followed by a medical degree from the University of Cincinnati. He completed internal medicine training in New York University, followed by fellowships in cardiology at Johns Hopkins University and the Cleveland Clinic. Of note, he also holds degrees in engineering, business, and law. Dr. Slepian's groundbreaking work over the years is truly and indeed inspiring, resulting in 250 published manuscripts in journals such as Science, Nature Materials, the Proceedings of the National Academy of Sciences, PLOS ONE, Circulation, the New England Journal of Medicine, and Cardiovascular Pathology. In addition, he has authored numerous book chapters, and his research has resulted in dozens of patents and a number of medical device companies. Two examples include co-inventing the first FDA-approved total artificial heart and the world's first biodegradable coronary stent. As an innovative teacher, mentor, clinician scientist, engineer, inventor, and real-world solution developer, Dr. Slepian has repeatedly brought national and international distinction to the University of Arizona. Dr. Slepian has served the University of Arizona as a member of its faculty for nearly 30 years. In addition to his clinical and research activities, he is also a dedicated educator. He has mentored and inspired hundreds of undergraduate, graduate, and postgraduate trainees, not only in the College of Medicine, but also in engineering and business management. In recognition of these accomplishments and more, he was named this past year as a Regents Professor, an honor bestowed only on 3% of the university's faculty. Please join me in extending a warm welcome to our very own Dr. Marvin Slepian as he delivers the 2020 Founders Day Lecture.
We invite you to share questions using the Q&A feature on Zoom any point during the presentation.
Good afternoon. I'm Dr. Marvin Slepian, and I'd like to thank Dean Abacassis for that magnanimous introduction. It's truly an honor and a privilege to be this year's Founders Day speaker. I wish I could be there in person with you. However, we're all adjusting to these trying times, and I hope that in the future, we'll all be able to be together in a more typical auditorium setting. I've entitled today's lecture, Viruses, Polymers, and Hearts, appropriate for COVID, M cubed, Mechanisms, Multidisciplinary, and MultiScale. And what I've attempted to do is follow in some of the traditions of past speakers, speak a little bit about my background and how that has influenced my approach to medical education, to research, and to clinical service. And I thought what I'd do today is to bring in some aspects which many of you may not know as much about me from my early background, because that has really set the tone for my entire career. And I will attempt to share that with you here this afternoon. Let me begin with some conclusions. Medicine actually operates on a multi-scale level, and I think it's important for today's investigators, for today's students, for all to consider that in addition to treating the patient, we have to consider the patient in relationship to their family and group, moving up, as well as certainly moving down at the organ, cell, and molecular level. I want to emphasize the importance of science. This provides really an opportunity to extend into new territories, new skills, and new perspectives. And it's important to begin early. This can have a big impact on one's career, as it has on mine, converting one from what we call a double threat to a triple threat. I will emphasize in this entire talk about the importance of mechanisms. This will provide better appreciation for pathophysiology, for difficulties in approaching diagnostics and therapeutics, and certainly the value of interdisciplinary work as well. In my lab, we look at things on the basic and applied level, but most importantly, we focus on translation. Not just bench to bedside, but what I call bedside back to bench, back to bedside. We look at preclinical as well as clinical things, and we have emphasized in a big way innovation, and I'll speak a lot about that. While we cover many different aspects and we are focused really on cell mechanotransduction, its impact on vascular cells, platelet biology, we also do a lot of work on novel polymers, and we've translated that, as I'm going to show you here this morning, into practical applications which can be utilized clinically, as well as in the area of mechanical circulatory support. And very interestingly, over the last few years, we have started a center at the university called the CABI, which is really focused on innovation and translation in a larger perspective. So what are the driving words for me? Immerse, explore, excite, involve, make it fun, wonder, curiosity, empower, mentor, question, learn to question, challenge, think, critically think, imagine, dream, envision, invent, create, and then make it happen. So how did this all start? Well, here's the beginning. I was born in New York, in Brooklyn, and I couldn't be at this state without having the appropriate parents who were just unbelievable. My father was a professor of medicine, and from a young age, I grew up in a medical house where literally the office was in the home. And my mother was an educator as well. always driving education. Yet they made it fun. And you may think that, well, that was a very kind of difficult, pushy environment. Not at all. It was a enriching environment where education and academics were made interesting and a challenge always. And uniquely in our home, we had space for a workroom and a dark room, which were the beginnings of getting one's hands wet. I was always interested in science and in books, but I was not just a bookworm. I used to explore the city. And as many of you know, I have a love for cycling. And living in New York City, great opportunities to hop on a subway as you became a little older to go explore things all around. I used to build things, was very interested in electronics, amateur radio, and I used to grow things. And I think these literally were the sowing seeds to getting me started in the laboratory. And, in putting this talk together, I reflected on some high school and some undergrad as well as elementary school science projects. And it was really my elementary school science projects that set the tone for my entire career. In the 5th grade, I did a project on blood. In the 6th grade, I worked on cells. In the 7th grade, I did a project on DNA. And ultimately, in the 8th grade, viruses. And I haven't strayed that much from that aspect, even in my own career. And then in the 8th grade, I built the heart-lung machine. So if you capsularize the work from the 5th to the 8th grade, I've only added additional layers as I moved forward based on that early work. A fun activity used to be to go to used bookstores. And in New York, at my friend's bookstore, I found this book, which really was instrumental in my career early on, Viruses, Cells, and Hosts. And I was hooked. I wanted to be a virologist. I had the unique opportunity through my dad and other friends to begin to work in a lab. And I have to thank Dan Amsterdam, who was head of microbiology at Downstate and at Kingsbrook, who on the weekends, on a Saturday, I would go up and he literally over a year period gave me a tutorial in the molecular biology of the gene. Hence mentorship. We're talking here in the 8th and 9th grades. And it was all about prokaryotic systems at the time, bacteriophage. I read this book and again was hooked. I had the opportunity in New York to go to, New York has these science high schools, to go to Stuyvesant. It's in Midtown Manhattan. You have to take an entrance exam, but it's an unbelievable school with fantastic teachers who provide enthusiasm and zeal to be involved in science. And I picked up this book on bacterial viruses by Stent, ironically called Stent, and really devoured this and moved on. I was interested in microbiology, virology. I developed a technique early on in high school of rapid identification of bacteria using a new technique at the time, electrophoresis, and saw the value of inventing things, of how you could come up with new solutions. Well, I was interested in genetic deficiency diseases early on. This whole molecular biology era. And having been at Isaac Albert Research Institute at Kingsbrook, they were studying gangliosidosis. And there was a student who had gone to Stuyvesant High School who showed when a virus infects a cell, it can pick up a gene and transduce the next cell, bringing that gene along. And that was Joshua Lederberg. And at a young age, he won the Nobel Prize. And not to be audacious, but I said, that's a good challenge. Could we not take the same strategy and do it artificially to create artificial transduction? And in order to do that, we need to create a synthetic virus. So I said, we need to really understand this system. We need to take it apart, manipulate it, re-engineer it. And during my entire high school career, this is what I worked on deeply in laboratories beyond going to school. I had the unique opportunity, being in Midtown Manhattan, of being proximate to many other great medical institutions. And literally, I went up to Public Health Research Institute, walked in and met several investigators, particularly Lauren Day, who probably said, who is this crazy science kid? And he allowed me to work in the lab. And I worked at Public Health for 2 years and met some unbelievable investigators, Dick Novick, Pernell Chopin, and really became immersed in viral systems. I learned some basic techniques, electron microscopy, and carry 60 stereoscopy to be able to look at particular molecules. We were able to prepare large volumes of bacteria and bacteriophage for analysis. And this really immersed me in physical chemistry and molecular biophysics, understanding molecular weights, size, shape, components, and being able to really characterize a large molecular structure. Ultracentrifugation, to be able to prepare large quantities of a virus, and uniquely to study its properties in terms of the sedimentation coefficient, diffusion coefficient, learning really classics of physical biochemistry and molecular biophysics. And at Stuyvesant, you had to be in the science fair. Mr. Baumel, the chairman, called me up and said, "Slepian, what's your mother's name? I'm entering you in the science fair." And this really started a transformational event. And as a junior, I described the biophysical properties of bacteriophage T5, including its molecular weight, characteristics, and components. But then beyond that, I developed methods, which I continued. And my senior year of high school, I really didn't go to high school other than one class, and I worked at Public Health. We developed 5 methods of making synthetic viruses, where we could package a range of DNA components and then demonstrate that we could actually transfect cells to effectuate a genetic change. We developed these large polyheads, which were a better technique to actually capture DNA and condense it, and then transfer thymidine kinase gene to cells. Needless to say, I was back at the science fair, and at this time, I won in multiple categories, literally 10 different things, including ultimately at that time in the Westinghouse competition. But I was the United States student representative to the Nobel Prize ceremonies based on this. Won an award from the Atomic Energy Commission, got to meet Glenn T. Seaborg, and also received the key to the city from Mayor Lindsay and from Jacob Javits. So you could say, well, this is unbelievable and could be mind-bending and mind-blowing for a young investigator. And I reflect back on this. You have to get involved in science. Get involved early. It can show that you can, if you understand systems, you can manipulate them. And it really comes down to mechanisms. But with hard work, you get somewhere. It's not about the awards. They're not the goal. But it's truly emphasizing that you can make an impact and make a difference. And that truly is the goal, to be involved in an early age. These were the drivers. Famous names in molecular biology: Delbruck, Watson and Stent, Hershey and Chase, Jacob and Minode. Interesting, Lederberg, Blumberg, and Zinder all had gone to Stuyvesant High School. So again, drivers of mentorship and by example. Yet I learned, even at that early age, of the importance of balance, of C.P. Snow and science and humanities. And that is so important, not only to be so focused only in one field, but to broaden oneself. I had the opportunity to go to Princeton and have great exposure there. I had some great mentors. I worked in Bruce Albert's lab early on, Uli Lemney, known for electrophoresis, Hal Weintraub. And there I continued on this molecular biophysics pathway, describing the geometry of viruses and understanding how quaternary structures can work together. In the summers and in my last 2 years, I also worked at Rockefeller and understood how tumor viruses were working. And this really was starting to drive my direction towards medicine and mixing virology and immunology. Ultimately, I did my senior thesis with Arnie Levine on the role of histocompatibility antigens and tumor antigens in development and neoplasia, that unifying hypothesis, and had the opportunity to work with Mark Kirshner and learn about microtubules and microfilaments, which today still are seminal in my own laboratory in terms of platelet function. Yet at the same time, Princeton had a way of broadening ones. Right across the street was a new foundation called the Robert Wood Johnson Foundation. And I had the opportunity to have in a course David Rogers, Walsh McDermott, Bob Lendon, and Margaret Mahoney. And they asked me to work for them. And I wrote a paper on the role of social work manpower in healthcare delivery. And this got me interested in policy and larger things. And again, the importance of balance, not only of science. So what were the lessons? Focus, focus, focus. If you go deep, you'll be able to really solve problems. It's really important to try to be the best in a particular area. On the other hand, there is a need for integrators with breadth too. And I've always tried to do this to create a dynamic balance in my own work. Work within your limits, but expand if possible between science and non-science. And it's useful if you can bridge the gap. And I've continued to do that throughout my entire career. I went to University of Cincinnati for medical school with 6 of my good friends from Princeton. I had the opportunity, again, it was research during medical school. I worked in Helen Glick's lab on platelets, and I've never looked back. Very seminal in so many mechanisms of disease, certainly in the cardiovascular space. And with Evelyn Hess, and they're connecting immunology and thrombosis. I defined, and this was one of my first papers, lymphokine-induced platelet aggregation, a novel lymphokine, which connected these systems. But beyond medical school in one place, and I encourage all the students who may be watching, it's valuable to have exposure broadly. And in my senior year, I went back to New York, knowing I wanted to go back for my residency, did ENT at Columbia, was a requirement at Einstein, cardiology, Sinai. Bellevue did infectious disease, and went back to the West Coast to try out Moffitt to see a great program. And then a miraculous experience. I spent a half a year at the NIH, which used to have these senior electives in immunology, rotating through 3 institutes, meeting amazing individuals who have gone on in their own careers to greatness. Broder, Waldman, Decker, Strober, Bill Paul in immunology, Al Steinberg. And if you notice on the bottom right, for 2 months, I had the most amazing attending, Tony Fauci, who at that time really was more focused on immunologic mechanisms and rheumatologic disease and vasculitis. But just as I was graduating, we read this ominous paper in MMWR about homosexual individuals with an unusual infection. And this is 1981's. And there were the drum beats. I went to Bellevue and NYU for my residency. It was a great place, tremendous experience, the largest hospital in North America, the new Bellevue, between the public hospital, the private hospital, and the VA. And uniquely, we didn't have separate unit teams. So you really became either a critical care physician or a cardiologist because you were always taking care of unit patients. And great exposure with Saul Farber and Frank Spencer. But there was a turning point. AIDS. AIDS went from being a unique, isolated experience to the plague that it truly was. And as chief resident, I described the experience at NYU, where more than 50% of AIDS patients died, and the wide range of opportunistic infections. And sadly, later at Hopkins, one of my colleagues was stuck with a needle and ultimately, as a resident, died of AIDS. And I knew as a virologist that wanted to have a laboratory that perhaps maybe this was a bit too risky to be involved in at that point, because there was another gnawing thing going on at NYU. Everyone had heart disease in New York. I think it may be the food, if you actually look at the corned beef sandwich shown here on the left. But I was involved in the Bellevue Sinai reperfusion trial in the cath lab. And again, this was transformative. So the light bulb went off, and I said, if I could take what I know at the molecular level and put this into cardiology, this might be a very, very exciting pathway. And that really was the basis for my career. I went back to the lab. In Dan Rifkin's lab, I started to work on myocytes, growing them for heart failure. And at the same time, I had the opportunity to conduct the trial. It was much easier in those days. I wrote to Sterling Winthrop. We obtained a drug. We had a local IRB, and boom, we were already putting swans in patients and rapidly looking. We could drop a wedge. But the era of the 80s was that drugs really weren't the answer for heart failure, but it was a great opportunity to understand the physiology. From there, I went to Hopkins in cardiology, which truly was the place at the time, with Mike Weisfeld and Bernadine Healy. And I were both of those fellows, particularly Bernadine. And there, clinical excellence was a given. It was all about research. What is the hypothesis? And this is so important in driving an academic medical career. Yes, that's me standing here with the other 5 fellows and the rest of the Hopkins crew. And on the top left is my good friend, Bram Zuckerman, who went on to be head of CDRH in the cardiovascular division at the FDA. And there in the library, read, think, understand mechanisms, pick up a pile of journals. Now you do it electronically. But if you read broadly and understand on a lot of levels, this is so useful. I literally lived in the Welch Library other than in the laboratory or on the wards or eventually in the cath lab. And I was immersed in new physiology. I, along with Lowell Maughan and with Dave Cass and with Dan Burkhoff, were involved in the early beginnings. I really was more working on the wet aspects and they were working on the more theoretic aspects, but I was learning rapidly. Again, I came from more of the science side. This was more the engineering side, and this rapidly became the BME experience, and I continued to do additional biomedical engineering training at Johns Hopkins to add in the engineering pathway, having been a chemistry and biochemistry major. And then stents came around. While I was interested in systolic and diastolic function, I didn't see it solely as a career, but I was good in the cath lab. And it was very clear that angioplasty and stenting would be an issue. So that's where if you have an early inventive thought and it's drumming around in your head, you can bring it back. And that's another teaching point here. And that led to this concept of polymer paving and sealing. Well, we all were doing angioplasty, but sadly arteries were closing. And in 1987, a paper came out by Ulrich Sigvard on the role of a stent. I spent the summer of 87 at Lausanne at CHUV watching Ulrich, and I quickly came back and realized that this was not the answer. We were dealing with metal in an artery. We were dealing with a time-limited process with a permanent device. We also were dealing with a cellular process with an inorganic material. We needed a better solution. I came up with biodegradable stents while I was still a fellow at Hopkins, but they were difficult to deploy. So again, this inventive step, why don't I take polymers and apply them as a thin film inside of a blood vessel to be a support, as well as a barrier and a means for delivering a drug. And this was the beginning of endoluminal paving, to address limitations of stents and to provide other capabilities in blood vessels. So what were we doing here? This was like back at the science fair, thinking about an unmet need and a limitation, thinking about things out of the box, understanding properties of different materials, coming up and seeing a solution. So pseudovirions were polymer, protein, and DNA a drug, whereas paving now was a synthetic material and a small molecule applied inside of a blood vessel. Same idea as the science fair, but many, many years later, hence the importance of early immersion in the laboratory. And we used interesting synthetic but natural type related materials. They were not novel polylactic acid or polycaprolactones. We borrowed from the plastics industry to thermoform. And I did this initial experiment in my kitchen when I was in Baltimore. I took a tube shown here as a mock artery with a catheter and an angioplasty balloon. I put a small polymer tube over it out of the material that I selected. I blocked the flow, brought in a little bit of heat. We deformed the material. It instantly set up. And next thing you know, we had paved the artery. We converted the tube on the left to the tube on the right. And hence endoluminal paving, which really was an early form of tissue engineering, was born. I went from Hopkins. I did a second fellowship. Angioplasty was just starting. There was no formal fellowship. So I made my own fellowship at the Cleveland Clinic, had amazing exposure to great individuals, as well as on the surgical side, and learned some of the newest techniques, which I ultimately brought to Tucson in terms of atherectomy, IVUS, and stenting. And again, great mentorship. I was back under the wing of Dr. Healy, at this point, who had been and was the president of the American Heart Association. And she encouraged me, write that grant. And my first grant in aid I obtained from the AHA, which was accelerating to my career, and paving really was born. But I needed better polymer experience. And I had the opportunity and the freedom in this second fellowship to shuttle back and forth. And I did a mini postdoc with Bob Langer in his laboratory, who ultimately is really the king of these type of novel biomaterials. And we worked together and have continued to work together from that point on. I came to Arizona, and really 3 phases, and many of you know this, so I'll go through this a little bit quicker. Certainly, the first phase was the typical of a triple threat strategy of clinical service, research, and teaching, aiming for promotion in an academic career to stay at Arizona. And I've been there now for 30 years. The second phase was take this experience and learn how to translate, and that's the artificial heart. And then the third has been a deep immersion back in the laboratory to now give back, to teach innovation, to do things in a larger level for the entire university. I've spent 28 years on the wards. As an original Bellevue chief resident, I never wanted only to do cardiology, and I stayed very involved in internal medicine, and it was the most fun time teaching and working at the bedside with many, many students and residents. And again, I reflect back on my dad, who I used to tag along with, and notice here, this is a homage to him. We're all wearing bow ties. This was bow tie day, and he's looking down and thanking me for it. Again, when I look at research, it's not only basic research or applied research, but it's translational research, bridging the gap. The translational process, to take a problem, see it, dissect it, go back to the bench, and then bring a solution back. And I've attempted to do this more and more. We've started a course through biomedical engineering where we bring engineering students on rounds to identify needs and work with us. I wouldn't be where I am today, and I couldn't be here without thanking the many great mentors at Arizona. From top down, Jim Dolan and Rube Bressler and Joe Alpert in medicine. Of course, Dr. Avey. Dr. Avey, I would always say he took a gamble on me that I would either be a flop or some of the research that I was doing would actually amount to something, and I hope that I made a difference. Thank you, Gordon. And Steve Goldman, who always understood and valued the importance of being in the laboratory, where I worked at the VA early on. And Bill Rusky, how to write a paper, make the figures. Frank Marcus and Carl Kern for being great colleagues. And Paul Fenster and Nancy Schweitzer, whose names, ironically, the labels are reversed, and for Monica Kraft today. And certainly on the surgical side with Jack Copeland, who really became a partner in arms, working together on the artificial heart, Gulshan Sethi, Francisco Arabia, and Rich Smith. Well, we moved paving forward. We quickly learned that we could apply these polymers and do this in Arizona, and we continued this research. We demonstrated that we could actually do this inside of coronary arteries. We built catheters to do this. We showed that we could actually do this with a laser in a blood vessel without heating the blood vessel. We could pave it, and we could locally deliver a drug, and the drug would go across the wall. We obtained patents on this. And interestingly, this wound up in the New York Times for making a big difference. But stents were coming. So even though we had a better idea, perhaps, it was clear that stenting was there. And we're now in the 90s, 1994 in the United States. And it was clear that we were developing restenosis. So we needed to go back to the lab. A stent would only approach the recoil in an artery, but we needed to work on the mechanisms. And the mechanism was thrombosis and smooth muscle cell migration and proliferation. So take that idea and go beyond. Let's develop a polymer that we can put in a blood vessel as a coating to block platelet activation and thrombosis on the site. And we did this working with Jeff Hubble. We synthesized novel materials, which could be coaters. This is the inside of a small animal artery. You can see this thin layer of polymer coating it as an exquisite barrier. And we showed that if you injure an artery on the left, you see thrombosis, but if you put the polymer down on the right, no thrombosis. And similarly, if you injure an artery mildly on the left, you get intimal thickening or restenosis. But if you apply the polymer blocking interaction of cells, you block restenosis, showing the importance of the mechanism of thrombotic interaction driving proliferation and migration. We published this in PNAS. We developed catheters to locally do this inside of actually large vessels in a man. And we thought we could combine this with a stent. And we showed that we could actually block thrombus on a blood vessel in a large animal model in a pig. And that by blocking platelet deposition, we actually reduced on the left the thickening or restenosis we would get without even a drug, just by taking platelets out of the equation. We then said, we need to go further. We need to add to this a means of delivering a drug to block these other mechanisms, an additive strategy. We could block matrix. And at that time, integrins were just really coming to the fore. And I felt it would be a great thing if we could block integrin matrix interactions. So we had to go back to basic science. In my lab, we demonstrated that smooth muscle cells expressed both beta 1, but when they were stimulated, beta 3 integrins. And if we were to block them, this is our hypothesis, we would interfere with their potential for migration. We came up with some novel peptides which could selectively block beta 3 integrins. And we showed in an important paper in Circulation that if we added a blocker of beta 3, we could block cell migration in vitro. And then we translated this using a polymer in large animal models in vivo. Again, translational research based on understanding mechanisms. So we had vision, and we're still on this pathway. We predicted that we would do more than just put metal in artery. We would biomanipulate a blood vessel based on these mechanistic strategies. This then led to the development of a technology which was commercialized. And this is a whole other aspect of my career, learning how to translate things. We founded Focal. This ultimately became a multi-use material, became a sealant. And we now use this in lung surgery. We also have developed this as a sealant for vascular techniques, and this has moved on. And ironically, our original biodegradable stents have become the backbone of what became Abbott BVS, which only recently was FDA approved. We developed techniques for enhanced revascularization and brought this to multiple centers. Here I am with Chris White at Oxnard doing some of the first myocardial channeling, which was another technique for angiogenesis. And we applied this to the outside of the heart as a new means of sealing and locally applying genes or drugs. And this is still in additional development. We brought techniques around the world. I had the opportunity to do angioplasty in multiple sites. I show one in Bangladesh where we did multiple procedures. And we've also advanced this to be able to do this remotely by developing robotic catheterization techniques. And we see the value of this in multiple other lumens as well. The next phase really was the artificial heart. In the second phase, one day, Jack Copeland came in my office, said, we need to move this technology forward. You know how to do this translational work. Well, we all know heart failure is on the rise. It's the final pathway of all forms of heart disease. And at the end of the day, you need a pump. And sadly, heart transplantation is not the answer. We only do about 2,000 or 2,500 in the United States per year. This began the Syncardia project. We all look a lot younger there, where Rich Smith, Jack Copeland, and I got together to move this technology forward into the world out of the university. And again, required multiple skills to move this technology forward. This was the heart driven pneumatically, and at times there are advantages that the heart provides, overcoming the failing heart, which otherwise would lead to a fatal situation for the patient. We designed a study. The hypothesis was that irreversible biventricular failure, a patient with this could be saved by placing in the artificial heart as a bridge to transplantation. We took patients that were literally dying, took out their heart, put in the artificial heart. At the end of when a human heart could be procured, the heart was taken out, but then, of course, they went on to live their normal transplant life. And we showed that despite the fact that these patients had an unbelievable mortality without receiving a heart, 70% of patients could be saved using this as a bridge to transplant. This had never been seen. And survival to transplant alone was about 80%. So this really was transformative, and mechanical circulatory support was born. And this came out and showed, this was the data we showed at the FDA, that without the device in orange, you had very poor chance of survival. But with the device, you had a very good chance of survival. In fact, you were on the heart for about 80 days and then went on to transplant, whereas the control group typically died within 8 days. We were lucky enough to publish this in the New England Journal. This received wide attention in the New York Times. The American Heart Association recognized this early on as a big achievement. We went on to show we could do this at other medical centers, not only at University of Arizona, though clearly it's one of the best places. We showed that we could use this in patients with cardiogenic shock. We showed that, and we fought the battle. This was part of my additional career of understanding policy and government. And we fought the battle not only with the FDA to receive approval, but with Medicare to receive coverage for this technology. We brought this heart around the world. Here we are in Berlin, training physicians on how to implant the device. Here we are in Holland doing the same. We also realized early on that, largely based on work in Germany, that we had a lot of experience in discharging patients with a modified small driver. So we showed that these patients, which otherwise had an 80% almost instant mortality, could at 1 year be out of the hospital 87% of the time using a device like this. And in fact, compared to biventricular support today, the artificial heart is more than double in terms of survival and remains the best strategy for end-stage biventricular heart failure. This is an example of patients in Germany on the artificial heart. Now, the good news, they're all alive. The bad news, they're tethered to a big driver. Well, we've been working on developing new small driver systems to get patients to be ambulatory. Here's a young individual who would walk around. This is one of our first patients on the portable driver, a young German policeman. And once he had the heart, he would, and the small driver, you couldn't keep him down. He would drive his car. He went back to work. He was really a great fellow. I had a great opportunity to meet him. And it really proves that there is a future for devices and technologies like this. You're going to see here a funny clip, perhaps here in a second. I don't want to advance it. Here he is coming up close. And what you'll see here is I used to joke because he used to like to go to McDonald's. And ironically, here he is. As a cardiologist, I couldn't condone this because it would cause coronary artery disease. But then you realize he really doesn't have any coronary arteries. So we used to tell him, go right ahead. Though, of course, he does have other vessels and he needs to watch atherosclerosis. Anyway, we went on to develop a small driver. Here we are with a latest version of this, and we continue to evolve this technology to show the ability of patients to kind of move out into the world. We've developed a small version of this now as a 50cc driver, and we've done almost 2,000 implants with this technology, and we continue to research this area. The next phase and the final phase I want to talk about is innovation. Well, I was doing this, and really under the guidance of Dr. Abe, he said, you're doing certain things that are really novel. You need to teach this. And through great mentors in the business school, I was encouraged to start a course. And I started a course on innovation, translation, and entrepreneurship. And medicine relies on innovation. And my mission was to take this body of knowledge of what I have learned and to really translate this to be able to teach this. The premise that innovation can be taught, to identify needs, to critically think, and to innovate at the interface. We teach a course over 16 weeks with 16 lectures. It's a full semester. And the idea that you take an idea and then you translate this and you implement it, and that becomes an innovation driven by unmet needs, differentiating needs versus wants versus demand. And you take a critical problem, you dissect it, find an aspect that you can treat, and then you go somewhere and you develop a solution. And you inject new science, omics, biomaterials, electronics. And we do a lot of exercises in the class, floatable paper, how the paperclip evolved, mechanisms. So the innovation space is beyond and between basic science and sort of a maniacal flight of ideas to come up and see a solution. But you have to go vertical to really do hypothesis-driven research to prove that it's going to work. So medicine operates on a multi-scale level, what I said at the beginning, and bringing this in is very, very important. I'm happy to say that I've taught this course for 10 years to 250 students across engineering, medicine, MBA. Many students have gone on to work in industry, to go to medical school, the FDA. During COVID, we worked on a Corona cuff for social distancing coming out of the class. I've received nice recognition. This is from President Hart and Dave early on at the beginning of my work in innovation, and then was recognized by the National Academy of Inventors as well. Here I am with James Wynant. for a lot of intellectual property and patents that have come out with the university. The final thing I wanted to talk about is the development of the Innovation Center, or COBI. The mission of the university is teaching, research, and service, but there could be more. We could take what we're doing and develop technologies which could move out of the university, creating things at the interface which will have value for society at large. So a COBI, the center that I founded, was developed to address an unmet need in universities, to move beyond. This was motivated by a grant from the NIH. I had the background having developed a lot of technologies and having spun them out into a wide range of companies. But you can't do this alone. So we create a network of multiple universities which now partner with us. And the mission is to be a creativity engine, to develop solutions, to find applications, to invent, to develop products, to help with driving grants, large studies, and ultimately to be a mindset change agent in the university and to drive novel innovation mechanisms. We've done this around Arizona, around the world, and broadly, and we couldn't do this without a great assistance. I think I'm covering up John Jackson, who's been on my right hand in really making things happen as the associate director of ACABI. We work on many broad areas, and because of lack of time, I won't get into all details, but come talk to us. We're working on novel materials, thrombosis, energy harvesting, wearables, robotics, etc. And many of the faculty have been involved, and we're here to help. And here are some that we've worked with broadly to move some of their own science and technology forward. And what are the opportunities? You only have to look at the top 20 diagnoses to figure out where we could go with this to advance technologies out of the university. And I have to thank those across campus that have been great supporters, particularly Jennifer Barton and Jeff Goldberg and Art Jamitro and many others, Dean Miller and Goes and Jones and Pierre Demier and Shelton and Durand. We're doing a lot more with this. We're working on stretchable electronics today, adding back to polymers electronics to make them smart. We've made smart balloons. We've been making smart paper, smart gloves. This is collaborative work with John Rogers. We live in a world where we are the wired well. Here we're developing head impact indicators. We've made smart tattoos. I'm going to go through this quickly just to give you a sense of things that we're doing. We're involved in motion sensing. This could be very useful in monitoring patients at a distance, particularly now in the COVID era. Motion is life. We've been working on ways beyond the Apple Watch. We've developed a biostamp, a small wearable technology which one could put on. And here I'm showing you that you can measure details of motion, which could be useful for medical diagnostics in many fields as well. And we've developed, I'm going to go through this a little bit quickly. We've been looking at activities of daily living. Here's walking. Here's sitting down. Here's brushing teeth. Here's washing a face, eating with a spoon, cutting with a knife, eating with a fork, drinking from a mug. You get the idea that if we could monitor someone at distance, of course, with secrecy and privacy respected, we'd be able to understand what's going on and take care of a larger group of individuals by using technologies like this. We've been developing other techniques which can measure blood flow. We can measure analytes as in sweat using this, which can become activated with a cell phone. And there are many others working on this here at the university as well. Here's a sweat study. We are using this to map signals in the heart. We all are familiar with the old EKG on the bottom, and you see on the top the new EKG. Again, the value of novel technologies. We don't even know what to do with this yet. We're developing biodegradable electronics, mixing electronics with polymers. And this was a paper of ours that was on the cover of Science. And we're working on smart paving layers, taking an old idea, making it new, and trying to advance this in a forward way. And certainly many know that in our laboratory, we're doing basic mechanisms of how platelets work, because again, thrombosis is key. And I don't have the time to possibly go into all the areas of research that we're working on, but we've made upheaval in the field. We've shown that some of the basic drugs that we use really don't work. And this is collaborative work with Danny Bluestein at Stony Brook and with Alberto Rodali at Politecnico di Milano. And we're developing and understanding the basic mechanisms of platelet activation today in seeing how shear and physics interact with cells to activate them. And we've developed a new model of the basic mechanisms of platelet activation. And with this, we've developed a new class of drugs. We call them mechanoceuticals, by which we change the mechanical properties of cells. And we were recognized by the NIH for this. And we received a major grant in this area, a U01, a quantum grant to really advance this field several years ago. We've been bringing people to Tucson with societies, and it's important to be involved in societies. I became the president of the International Society of Mechanical Circulatory Support, and young investigators involved in this. President Robbins came to this when he first came to the university. And we had the World Congress on Mixed and Augmented Reality here a year ago. And again, we had the World Congress on Biofluids. This was probably the last Congress that people traveled to. This happened right before COVID, and this was held in the Innovation Center here. I've been involved in national societies and beyond with the National Academy. And I was elected to BEMA. And this, again, is the interest of policy and government and science. And my motto has been, you need science, medicine, engineering, and business to innovate, but you need law to operate. And many of you know, I ran a meeting for the National Academy on law and medical device development with colleagues from University of Arizona. And there's Dean Miller and Tara Sklar from the University of Arizona, Chris Robertson. And then next thing you know, I wound up in law school. I'm doing this to gain additional knowledge and be a student as well. To keep the brain sharp, you have to always continue to learn. And it's been a great experience. We've developed the Regulatory Sciences Center to, again, understand how to move science and technology forward in a big way. And I had the privilege to work with Professors Massaro and Pideaux, even in the pandemic, to provide some guidance on constitutional norms for pandemic policy. I couldn't do this without many in the laboratory. It's all about the students and exchange students from Italy working with us and young students that I've mentored in the laboratory. Every summer, we have the summer bioengineering group that I mentor. And that's where I am most fun. Being with students, in front of students, teaching them, immersing them in the laboratory, working closely. And as I get to this point in my career, while there are many great clinicians, and I would not give up for a minute being involved in clinical medicine, but having the ability to work with young people, that's how I got started. And that is the enthusiasm that I want to convey to students to get involved early on. We always have students in the laboratory in the summer. Here's some of the students in my lab currently. And we've been driving senior engineering projects. Here we're looking at platelet stiffness. And we're looking at sound analysis and image analysis and virtual reality. We've been driving these teams and supporting them through funds that we have for Akabi to drive student teams. We've supported for the last 5 years, 25 students a year in this type of work. And they built great things. Here's a smart sensor as a foot insole. Here's a team working on a point of care thrombosis system, virtual reality for CPR. Here's the team doing this, another microfluidic group. And it's great to see our students succeed. One of our teams won first place in the Innovation Challenge for Engineering. And one of my students was the graduating senior with the highest average and run awards for that, the highest graduating senior in biomedical engineering. And to see them get jobs, now working in mechanical circulatory support for Abbott. And one of my students now getting her PhD and multiple PhDs now coming out of an MD's laboratory, which I think is exciting. Or getting first place in a poster competition, or seeing students from my laboratory move on to go to medical school. The final thing I was going to show is what we've been involved in COVID. We've worked through the Innovation Center on building ventilators, a basketball ventilator for Arizona, using either a foot pump, a multi-volume or pressure system, a portable system. Working with the University of Liverpool in England, we worked on novel face shields, which we've now provided to many in cardiology. We've worked on a robotic access robot. We call it Rambam, after Maimonides, of being able to reduce the need for PPE and access patients without exposing healthcare workers. We've worked on a system of providing materials in a forward way with Mike Jacobs, a former graduate of University of Arizona. We've developed an early technology. This is with Sai Parthasarathi, a non-rebreather technology through Akabi. And this was spun out to create Ciox, named after Sai, a company working on novel rebreathing strategies for enhanced utility in COPD, but certainly has a role to prevent viral spread in COVID. And again, working on wearables. I mentioned the Corona cuff. And in ECMO. This year, I'm the president of ASIO. And in ASIO, we worked on developing an action portal. We created a guidelines, which we nicely had published in both the ASIO journal and in Circulation Heart Failure. I thank Dr. Schweitzer for facilitating getting this in, but peer-reviewed and rapidly out. We described the first large series of ECMO in patients with COVID. And it's nice to know recognition when you see your paper now in up-to-date, several of our papers, as the guidelines for ECMO in COVID. This is the database which we continue to enroll in for COVID in ECMO and in renal disease. And we've been working on the thrombotic complications in COVID in terms of pulmonary emboli and the devastating thrombotic complications that occur with cytokine storm. We recently described a signature to differentiate whether it's platelet activation by virtue of ECMO or a device or the inflammation. And this was nicely published in Thrombosis and Hemostasis just a couple months ago. I have to thank Dr. Roka Moya, who's been a star in our laboratory to move these technologies and this basic science and recognition forward. And we have now a new way of diagnostically differentiating this. And this became a novel intellectual property for the University of Arizona to be able to figure out what is the driving force clinically. We're also, again, back to Motion is Life. We are working, this was just 2 days ago with the Arthritis Center. We all recognize that we are being shut in. In fact, in the New York Times on Wednesday, the word of the year was lockdown. It actually beat out coronavirus with this imposed inability to move around, restriction of mobility. And again, we're using motion sensing in a collaborative way, using a cell phone with a technique we're working on with University of Nebraska collaboratively to develop a technique to be able to measure, quantitate, and train and maintain flexibility and diagnose individuals at home as well. Here are some of our medical students working with this to define flexibility as well as to define the neuroexam. We're doing this with Dr. Bruce Kuhl. And our goal is to develop a system of integrated distance access technology in the new way of medicine, which is not going back. Telemedicine, telehealth, the work that Dr. Weinstein and others have been working on, and we're working on with sensors, and many others at the University of Engineering are working on, will be the way of the future. We need to put this into our curriculum as well. Of course, I couldn't do this without a supportive family. My wife, Donna, my daughters, Rebecca and Sarah. And here, they both, Sarah and Rebecca are proud graduates of the University of Arizona. And hopefully in the future, my daughter will be going to medical school. We hope, hopefully at the University of Arizona at some point, or at any one of the other great medical schools in the United States. And again, the students in my laboratory, in other laboratories that we are adjunct with around the world, helping us. And we couldn't do this without great funding from the NIH, the American Heart Association, private individuals, foundations, and others to make this happen. So it's all about the students. I want to thank everyone for listening. The takeaways here are: get involved early. Think about mechanisms. Understand how to question. Ask the right question. Frame the hypothesis. See the solution. Innovate at the interface. And keep innovating. Thank you very much for your attention.